All right, welcome to this week's meeting. This week's meeting, I had some problems with encoding the recording, so I'm going to give an update on what we did today. And I want to give an update on some of the work that some of our aspiring GSoC students are up to as well. In fact, that's most of what our meeting was people presenting, and that's why I wanted to give an update. And so we're going to talk a little bit about hypergraphs, and we're going to talk a little bit about topological data analysis. So we have two students interested in these two projects, and so we won't be finding out who is being selected for the project until the beginning of May, but we are interested in people contributing to this project. This is the DevoGraph project throughout this summer. And so let's get started. So we'll start with the Hypergraphs project, and I have some screenshots from the recording. The audio didn't work, but I did capture a lot of the slides that people made. So our presenter was Mehul Arora, and he's been joining our group for the last several weeks. And he's interested in this hypergraphs paper that our group put out at the end of last year. And this is hypergraphs specifically for developmental biology. So he was very intrigued by that because he had written himself a hypergraphs paper, not on embryos, but on human brain data. So this is a nice, he's going to try to transfer these skills over to development, but this is also nice that people have expertise in these areas that are synergistic with what we're trying to push forward. So Mehul wrote a paper called HyperGALE, and this is hypergraphs, and then the acronym GALE. So GALE is Gated Attention, GA, and Learnable HyperEdges, LE. So this is a classification of these structures. You're using two things. You're using gated attention and learnable HyperEdges. So this paper that he did, he was an author on, it was an attempt to sort of get your hands around autism spectrum disorder. So this is based on neuroimaging data. And what they're going to do is they're going to characterize brain activity based on different types of autism, or try to classify different types of autism. And so this is the standard boilerplate for a brain imaging study. The important points here is that autism spectrum disorder is heterogeneous. So there are a lot of different symptoms, a lot of different severity levels, and it's very hard to just make the diagnosis by looking at the behavior or by just looking at a set of images of the brain. And so we want to be able to use machine learning, specifically graph neural networks, to come up with a good diagnostic criterion. So we also can link autism spectrum disorder to distinct patterns of functional connectivity. And so functional connectivity is a type of connectivity they use in neuroimaging. It involves making assumptions about what brain areas are connected to what brain areas based on function, or what voxels are connected to different other voxels and other brain areas based on function. And so this is one of the things about this model, is that he's using hypergraphs as a way to sort of characterize the regions of interest. And so those regions of interest are things that you might parcel out of whole brain data, such as like the hippocampus, or certain regions in neocortex, or certain regions in cerebellum. And so you can actually do this with post-processing, and it's this region of the brain. But each region of the brain, of course, contains a number of voxels. And the voxels, of course, are the standard minimal unit in fMRI, for example. Those voxels then serve as the nodes, and then the ROIs serve as the hypernodes. So each hypernode has a bunch of nodes in it, and each hypernode has a hyperedge to another hypernode or another ROI. And in turn, all of the nodes within the hypernode are all connected to other nodes within other hypernodes. So you have these hyperedges, which are sort of averaged versions of different edges that go from the edges to other edges, and depending on their category or classification, they either are, there's a lot of variation there. So this is a thing that we're also interested in. with respect to developmental hypergraphs. And in developmental hypergraphs, we use actually a more formal method of category theory to determine the different categories. So this is something that people have used novel machine learning methods for, classifying ASD subjects, but also working with these types of multi-level data. And of course, I guess if you want to call it hypergraph neural networks, that serves our needs very well. And so they went through their data set and their pre-processing steps. They talked about the HyperGALE model. So HyperGALE is a novel hypergraph-based model, and it's being applied to ASD classification. And of course, GALE stands again for Gated Attention, GA, and Learnable HyperEdges, LE. Then there's model evaluation, so they're using different methods and comparing it with graph-based methods, which I like because I think we need to do a lot of benchmarking of our GNNs, especially in terms of the kinds of results we get with DevoWorm and some of the deep learning methods there, but also other methods of classification on the other end. So when you have your model of hypernodes and regular nodes and hyperedges and regular edges, can you get a better sort of result using something like a deep learning technique or a transformer, just a plain vanilla transformer or something else? So this is good. This is good that we have this sort of interest in doing these benchmarks. And so they did a pretty decent job in terms of accuracy and area under the curve. So this is, of course, the standard model evaluation. And importantly, they did focus on interpretability and looking at the difference in biomarkers for different types of disorders. So, one of the things about biological data, especially developmental data, is we have this issue of interpretability. A lot of times we use models especially for large data sets or what we call big data, and we don't really understand what we're doing. So in a sense, what this hyper network model is doing is it's reducing the dimensionality of a network. And so you have these hypernodes that sort of take in a bunch of nodes and they're hyperedges that sort of average out the edges. So basically is a way to interpret, say, like a complex network. And complex networks indeed are a little bit hard to make interpretable for people. People have their intuitions about what's going on, and they plot them out in a network, and you get this hairball, and it's not really something you can say very much about. Sometimes when we do things with genomic data or gene expression data, we have issues of using dimensionality reduction or multi-dimensional methods that give us a visualization that looks very pretty, but it doesn't do very much in terms of interpretability. Oftentimes people will see something and say it's a cluster, and it's not really a cluster because the standards for or the support for a cluster are not there. So I think this is another very important focus, is interpretability. So this is the figure that he kind of showed about their modified hypergraph convolution. So they're doing this series of convolution steps to determine the connections between the hypernodes and other hypernodes with hyperedges. So there's the HyperGALE algorithm down here, and it's hard to see because of the way Jitsi displays it. But basically, you work from these parcellations of data. You move that, you get a measure of functional connectivity, and then you get all these different transforms. You use a ReLU function, and you basically get these networks. So basically what you're doing is you're growing these networks from the best fit to the data. And so this is the results, comparing non-graph methods and traditional methods with the graph-based methods, transformer-based methods, and then their approach, which is our hypergraphs. So this is of course, there are other hypergraph models in the literature. HyperGALE then actually does a really good job, actually higher performance than any of the other ones. So in terms of accuracy, I think in terms of area under the curve as well. And so this is sort of, they've done a pretty good job in terms of the hypergraphs, making this a good analytical procedure. And of course, our focus has been more on theory, but they really have shown that this can be used quite well for at least for neuroimaging data. And so this shows some of the experimentation. So what they're doing for some of this in terms of looking for HyperEdges, they're using this KNN, or these k-means procedures to sort of get cluster things and find like edges and make HyperEdges out of them. So they're using this type of classification technique, and they're running this and getting results. And so suffice it to say that you have different issues with HyperEdges. They're not like obvious things. You have to search for them. You have to classify them based on what the ROIs are. And so this is a problem too with some of our developmental data. So sometimes we have these easy to understand labels. So like in the paper that we did, we talked about some of the tissues that exist. And of course, in C. elegans, you have individual cells that have a label, and they divide and differentiate deterministically, meaning that we know what their fate will be. We know what the lineage tree is. We know what each cell is in the lineage tree, and it can be labeled, and it's reproducible across individuals in a species. Now, in the case of other biological data, perhaps if you're looking at human or mouse development, and you're looking at different tissues being formed, the cells are, they respond to signals. We don't have like a label for every cell. And oftentimes there are these processes where whatever cell gets the signal first is the cell to differentiate. Now, those processes are predictable, but my point being is that they don't really have labels that we can use. So even like functional labels aren't very useful when you have cells that, differentiate depending on certain signals, or de-differentiate, or they go through apoptosis or whatever. So it's very hard to get a good sense of what a cell, what a label of a cell is, or what it even means. So this is one of the challenges of developmental data in the neuroimaging world. You know kind of what your regions of interest are, but you don't necessarily know what's inside each voxel. So that's a little bit of a limitation. But you know that those voxels are in an ROI, so it makes sense that you know kind of what their function is. In development, you do have a similar thing, but you often don't have labels. You have functional labels that we've gotten through annotations, but those annotations can be very high level and sloppy in terms of giving you a good sense of what kinds of transitions are happening, what kinds of categories you can build, and so on. So if you go back to our paper, we use organisms, we use cyanobacteria, we use some sort of analog of C. elegans, and so it's clear that there's the spatial component, that there's this temporal component, and that you can label cells and tissues. But the thing is that in other organisms or other systems, especially when you're trying to get things out of the data, it's less clear as to what those labels are, or what those labels should be, or what should be in these hyper nodes, because you're going to have a lot of different cell types in a single hyper node. So like, for example, the liver, the human liver, or any mammalian liver, has a lot of different cell types in it. And so we kind of know from estimates what the distribution of cell types should be in there. But quite frankly, those labels are a little bit idealized. And so those are some of the challenges of mapping data to these kind of methods. And this is something that we will encounter in applying hypergraphs to biological, especially developmental data. Now, they talked about interpretability specifically. So one of the things they're trying to do is they're looking at ROIs, and they're noticing there's a huge overlap in ROIs. So you can have ROIs for something like decision making, which is an important part of autism, to understand that network. But the thing is that those ROIs overlap. So that means that you're going to have nodes within maybe two or more hypernodes. Because if your ROI represents a hypernode, and those ROIs overlap, then those hypernodes overlap. And so you're going to have nodes or voxels that are in more than one ROI. So this is something that, actually you can identify through gated attention, but is a problem for sort of our standard model. And so this is interesting because it provides both a challenge and an opportunity. The challenge is, of course, understanding that you have to have a model of overlapping hypernodes. But the other thing is that you can use techniques like gated attention or some other technique to actually identify trouble spots or places where you have this overlap. And so we can address it in a way that's, makes sense in the model. And so, we can find individual differences in connectivity and all that. We have this here. So we have this comparison of typically developing brains versus autism. And of course, those different conditions yield different network topologies as we might expect. So we can observe that, but we also can observe that across subjects. It's not surprising. And of course, you'll see this in development as well. Even in C. elegans, we have, wild type development, and then we have development that arises from in specific mutants. So sometimes specific mutants will be missing some cells, or the tissues won't develop normally, and so we have to account for that. So using their method, they were able to identify key functional networks. And again, this is based on neuroanatomy, so we know from neuroanatomy what these networks should look like, although some of the regions of the brain are always kind of in dispute, like the limbic system, which is hard to really define in terms of what areas, what ROIs are all involved in it. You have executive function and attentional networks, and again, attentional networks can include a lot of things or very few things. It's one of these things that, you can toss things out and get a better result than if you include things, but then you get a misleading result. And then, of course, the default mode network, which is also, again, another thing that is very hard to clearly define because we don't really know we kind of know what it does, but there's been this debate in the literature about it. So all I'm going to say about this is that our networks, especially when we start parsing it out by function, can be very difficult to define. And sometimes the results can be misleading. But, and of course, the overlap as well. So that's another challenge. And so they were able to find, different differences between the autism brains and the typically developing brains. And that's, I guess, the important point. Now, this is an illustrative of the gated attention module. So 2 images back, Himanshu raised his hand and he asked about this. He didn't see a specific mention of the gated attention module. So they're using machine learning version of attention, not the attention network from the brain, but the machine learning version of attention. And they're using this gated attention module. And this is the diagram of it. So basically it learns this alpha parameter iteratively, and it learns it from an equation. And then the alpha vector is multiplied to get the final node features, which is fed to the readout layer. So basically you have all these alphas here that are estimated, and then those are combined into this alpha vector, which is then multiplied out here with the estimator, and it gives you this readout layer, which is z equals this term. So you get this, you get this gated attention where I guess it's selecting things, and then it's using this estimator to tell you what the answer is. So this is a very, this is kind of a breakdown of the model. I'm not going to get into it too much because I don't know if I really understand it enough to say exactly, to give you the ins and outs of it. But this is a transform that's been used, and there, of course, there was the paper, All You Need Is Attention, which is something that I think is both very arrogant, but also very maybe true. That attention has a very important role in most machine learning activities. So here we were after this talk, and it was a very good talk. Shurvraneel, who is also in the meeting, asked this question: while researching, did you choose the number of neurons later of the MLP layers, which we saw in the gated attention module? And I think that Mehul answered it pretty well, that sometimes, we think in deep learning that more layers are better, but in fact, in the case of gated attention, more layers are not always better. So that's a nice insight, and it's actually kind of a divergence from the typical, what we think is like the recipe for optimal deep learning. And so that's good. And then Himanshu, of course, asked about the gated attention diagram, which was shown in that last slide. And then Susan apparently was having trouble with her bandwidth. So thanks for all the fish. And that was Susan for today. And Morgan, of course, is really interested in both neuroanatomy and deep learning and machine learning. So I kind of hope that Morgan has some feedback. I know he was kind of came in the middle of the talk, but I'm hoping that he has some insights for us as well. And this is especially true if Mehul gets selected for GSoC. So this is the HyperGALE paper. This is in the arXiv. It's arXiv 2403.14484 version 1. And this was just put out like, earlier this last month. So the 21st of March. So this is HyperGALE ASD Classification by Hypergraph Gated Attention for Learnable HyperEdges. So again, they're using the gated attention module to try to construct hypergraphs. They're learning the hyperedges, and we're not just kind of assuming the hyperedges are, connecting to hypernodes that we know are kind of associated. And we're using our functional connectivity to sort of give us these potential hyperedges. So one of the things they did in terms of their data set, they have this ABIDE2 data set, which is a data set of neuroimaging data. So again, your data set is going to be important for these projects, and we'll talk in a little bit about some of the data sets for the DevoWorm project. And so from the abstract, HyperGALE not only improves interpretability, but also demonstrates statistically significant enhancements in key performance metrics compared to both previous baselines and the foundational hypergraph model. So this is interesting that, there's this foundational hypergraph model, but there are also other types of methods. And we have these baselines that we can use, and they outperform those. So I'm hoping that, maybe hypergraphs are sort of the key to our efforts in DevoGraph, and especially in tackling some of these developmental data sets. The advancement HyperGALE brings to ASD research highlights the potential of sophisticated graph-based techniques and neurodevelopmental studies. So this was actually neurodevelopment in like the human brain in not infancy, but in childhood. So this is something that, of course, there are similarities because you have this change, this plasticity, but it's a little bit more fundamental in our case because you're actually getting cells being born and being connected together, and you get a lot of features in our data sets that really require these kind of models that expand outward. And we're going to talk about that in a bit too, where they expand and they take in new nodes and they wire them in a certain way. And then they have these hierarchical organizational levels. So you might have your nodes, which are the cells, and then you have tissues, and then you maybe have networks like connectomes. You might also have things that become synthesia, like muscle, which means that they have multiple individual cells and they get fused, and you have multiple nuclei and other things like that. So we have a lot of things that we can actually draw from that actually pose unique challenges to these kind of methods. And so this is their sort of pipeline here where they have the ABIDE2 data set, which is the autism brain imaging data exchange. Then they have the Schaefer parcellation, which is the technique they use to get the ROIs. So you parcellate different parts of the brain, and there's a whole map for this, so you can align the data, and it's very very formalized compared to what we have to do in embryos. Although in C. elegans, people have done a lot of work on sort of standardizing, segmentation for C. elegans cells. So this shouldn't be too hard of a problem in C. elegans, but in other organisms it might be a little harder. And then finally, your functional connectivity matrix, which is the standard measure of functional connectivity. And then, for the HyperGALE pipeline, you're creating hypergraphs from this functional connectivity matrix. The hypernodes are these parcels, or these ROIs. The nodes are, individual cells in this functional connectivity matrix. The edges are these connections, or where there's a correlation or covariance in the functional connectivity matrix. And then the connections, the hyperedges are actually discovered through different algorithms that are applied to that for that purpose. And so, you can imagine if you have two hypernodes with a bunch of nodes inside, and they all have different connections and different weights, it's going to be hard to know what the true hyperedge edges. So sometimes we have hyperedges that are like more likely or less likely. Sometimes we'll have multiple hyperedges that kind of converge on one area. And, it tells us information that we wouldn't know just from building like this hairball, which is the typical model of a network. This hairball, where it's hard to interpret what's going on, but you also have a lot of just kind of, places where there isn't a lot of structure on the graph. And there are ways around that. You can do things like community detection and looking for different motifs, whereas we'll talk about later simplices. So then there's hypergraph convolution, which is what we use for this convolution step, then gated attention, and then the MLP readout, and you get these two different measures here, the regular brains and the ASD. And so Mehul brought up another paper in our discussion. This is called Attention-Based Deep Multiple Instance Learning. So this is actually something that's, kind of their attention methods were based on the gated attention method in particular. So this is a paper from 2018. This was also on the arXiv. And so the abstract reads multiple instance learning, which is MIL. This is a variation of supervised learning, where a single class label is assigned to a bag of instances. So this is like a bag of words, where you have your instances in this sort of Polya's urn, and you sample each instance, like you would sample each word in a bag of words. And they're kind of nominally independent, but you can replace them, sample with replacement or just sample them. And it gives you a lot of power to sort of randomize what you're drawing from. So you're dealing here with labels and class labels. And so this is an important point here, because, the way we treat our sort of classes, sometimes we have labeled data and sometimes we don't. Like I said, sometimes the labels, though, kind of fall in this in-between area, which is in between being very reliable and not reliable at all. So if I say something is muscle, it's maybe muscle at some point in development. It's not muscle from the very beginning. You might have a connectome. When do we start to call it a connectome? Maybe when we get two neurons connected for the first time. To know where that boundary is, to know where there's actually some structure that would resemble what we're trying to do, which then translates to the classes and the class labels, is going to be a hard problem. And so you're going to have to do things where, you do things like multiple instance learning, or something else where it's not the obvious choice. So in this paper, we state the MIL problem is learning the Bernoulli distribution of the bag label. So this is where you have this distribution within the bag, where the bag label probability is fully parameterized by neural networks. So you're building this distribution of labels within a bag, you're sampling blindly, and you're getting, they're nominally independent. So these are, you're kind of choosing a label from this distribution. Furthermore, we propose a neural network-based permutation invariant aggregation operator that corresponds to the attention mechanism. So this is like this attention-like mechanism. Notably, an application of the proposed attention-based operator provides insight into the contribution of each instance to the bag label. We show empirically that our approach achieves comparable performance to the best MIL methods on benchmark MIL datasets. It's this multiple instance learning. And it outperforms other methods on MNIST-based dataset and two real-life histopathology datasets without sacrificing interpretability. So this is, an interesting point here. So basically, they talk about this here, where you have this scenario sometimes in real-life data where you have multiple instance learning, where you need to learn from a weakly annotated data set. So again, this is exactly what I'm talking about with the developmental data. It's weakly annotated. It's not, the annotations aren't great, but they're not totally worthless. And so, this is something we have to work around. So the problem with weakly annotated data is especially apparent in medical imaging. So citation Quellec et al. 2017, and where an image is typically described by a single label. So, sometimes you have images that represent benign tumors or malignant tumors, so you classify them in that way. But there is other information in there, and in fact, those labels can have you can have false positives, you can have true negatives. So the labels are based on like some, criterion that you pick, like some set of features. And if the label is weakly describes what's going on, so a lot of times you get a medical diagnosis where the symptoms point in different directions, and so the features maybe give a mixed signal. They don't all point in the same direction. So that label of benign or malignant is going to be harder to make, or it's going to be harder to say what data fits into those categories. Multi-instance learning deals with a bag of instances for which a single class label was assigned. So this is again where we're choosing our labels. We're drawing from a probability distribution. It's not blind, but it's based on the available information. Hence, the main goal of MIL is to learn a model that predicts the bag label, which is a medical diagnosis. So in the bag, you have a bunch of labels or medical diagnoses given some set of conditions, and you draw from that distribution, and you're able to get maybe the right thing. So then an additional challenge is to discover key instances, or for example, the instances that trigger the bag label. So there's certain features that may be very powerful in making that diagnosis. Those are key instances, and finding those is the important thing. In the medical domain, the latter task is of great interest because of legal issues and its usefulness in clinical practice. And so they talk about this bag classification. In order to solve the primary task of a bag classification, different methods are proposed, such as utilizing similarities among bags, embedding instances to a compact low-dimensional representation that is further fed to a bag-level classifier, and combining responses of an instance-level classifier. So only this last approach, combining responses of an instance-level classifier, are capable of providing interpretable results. So, we have these different techniques that we can try. And so, I'm glad that Mehul has put some effort into this, finding out what these different options are. Okay, now we're going to move on to the next presentation. So Pakhi Banchalia is another GSoC hopeful, and she's actually working on this problem of DevoGraph from a different angle, and that is topological data analysis. So Himanshu has signed up to be a mentor for this year's project. So he'll be co-mentoring the project with me. And he's been involved with 2 other GSoC projects that I've been a mentor for. So in 2022, he was on this open source sustainability project, and he built a wonderful reinforcement learning model. And then he worked on DevoWorm last year, and he actually worked in DevoGraph in building topological data analysis, part of that pipeline, and some of the techniques that Pakhi has been looking at and thinking about going further and making improvements upon that. So Pakhi showed us her notebook, and so she was actually looking through a lot of Himanshu's code and looking at how it can be improved. She's also been looking at 2 other models, Neural Developmental Programs and another associated topological data analysis method. Last time we talked about Neural Developmental Programs, this was 2 weeks ago, where we had this idea of the Neural Developmental Program. So basically, this is a technique where you can grow neural networks. We can, each one of these nodes in this graph that we see here is a neural developmental program. So this is very much like sort of an evolutionary algorithm, but it's not an evolutionary algorithm. It's called a developmental program, so it does a lot of the same things that an evolutionary program would do, but it does sort of this developmental aspect where it's born and dies, or it changes its wiring so it exhibits plasticity. And then, of course, you get this network of nodes that are wired in these, what we'll call simplices, which we'll talk about in a little bit. But these are, these like things like triangles, or, other types of basic shapes that you can identify within a network that are based on the connectivity. So in a neural developmental program, you have graph convolution, which is where you try different connections and you see which ones are the best fit. So in this case, we have a triangle, or a 3 simplex, which is a triangle, and that was a good fit for this data. And then, we have this transformation from one time step to the next, and we do this convolution. Then we have this growth phase where we actually take the graph state, which is defined here, and then we move to the next time step, and we get this S hat, which is the estimator, and that graph state is updated via local information aggregation. So we're aggregating information and we're growing the network. So this is graph growth where we add two nodes and we have to connect them in to the existing programs or nodes. And then we do this weight prediction step where we're predicting the edge weights updated as we go along. So this is actually something that is Sebastian Risi and his collaborators have worked on. So this is so that means that there's an open source repo for this. But this is something that we could use in a lot of different ways, not just in DevoLearn and DevoGraph, but also in some of our agent-based stuff in the orthogonal lab as well. So this is, this iterates to a new development cycle. And then, of course, this is based on a paper towards self-assembling artificial neural networks through neural developmental programs. We talked about this 2 weeks ago, but I think it's worth talking about again because she's going to be using part of this as part of the project. But then the second part is this topological data analysis mapper. And so this mapper is actually an interesting technique. And so the best way this is based on a paper actually back from 2007 called Topological Methods from the Analysis of High Dimensional Datasets in 3D Object Recognition. This is the first mention of a TDA mapper. So this is a standard tool in topological data analysis. And again, this is the second part of this proposed project, and it's an open source repo, so this is something we can fork and use. But Pakhi actually created a demo in Mathematica, which is kind of interesting. Now, this is a very generic demo, so this just shows these different graphs as you move them around, and they kind of have this sort of physics here, this force layout where it's kind of moving against, you can't connect them like this. You have to kind of, so they're independent little modules that don't connect, but they kind of move against one another. This is an interesting set of things going on here in this demo. And what's interesting about this is it's generic, and you can create this. This is actually a UMAP projection where you have these nodes are connected, and you can create graphs. And so you can see that there's a small demo here, but it doesn't really look like anything biological, maybe a little bit. But it's actually giving us, something we can work from. So if we want to think about this in terms of developmentally specific data, there are a lot of networks in the embryo that kind of emerge from maybe different tissues forming or different parts of the embryo differentiating, maybe basic polarity, or maybe like the animal pull, where you have animal cells at one end of the embryo. And so you can actually model all these things with these kind of networks. And this is, of course, the TDA mapper technique. And so this is, a demo that you can work out here, but it doesn't really look like anything developmental, but we can, put the flesh on the skeleton, as it were. So that's a nice demo. But this paper, got back to this paper, we actually have some interesting points here. So this is the method where they first proposed this mapper method. We present a computational method for extracting simple descriptions of high-dimensional data sets in the form of simplical complexes. So these are the things that in this demo, this is a simplical complex here, these triangles, you have diamonds, you have other shapes that form, and so those shapes are like 3 simplices and 4 simplices and things like that. And it allows you to break down your network into components that you can then identify as like motifs. So like, if we know that there's some developmental process happening and there's, things are being connected together into networks, we can maybe when we see the first emergence of triangles, then we know that we've reached a threshold. We can put a label on the data as opposed to before then when we couldn't. So as cells divide and then differentiate or come into contact with one another through migration processes, or as the embryo expands the number of its cells, you start to get these connections that are that can be defined by these simplices. And so that's a very important tool, I think, especially in these growing networks where you're adding nodes into this network. And you start to get features that emerge. And so at a certain time, we can make a statement about, like, when this thing actually has a label versus another label. A very interesting kind of approach. So this method is called Mapper. It's based on the idea of partial clustering of the data. So in the last technique, we had this k-means clustering, and we had this search for different HyperEdges. In this case, we're doing partial clustering of the data, guided by a set of functions defined on the data. So this is a different approach, and now I'd like to see, if, say, both of these students get selected for the project, I would actually like to see, some sort of communication there, or dialogue between what people, what we're doing with partial clustering versus clustering on, maybe something like HyperEdges, and make comparisons, and maybe develop maybe a new tool, maybe, know which one is better for whatever, there's probably going to be different conditions under which one method is better than another, and getting that all worked out. The proposed method is not dependent on any particular clustering algorithm. So this is interesting that this mapper doesn't require a specific clustering algorithm. In this demo, actually, we're using UMAP as a visualization technique. So, this is something that we don't have to use UMAP. We can use another technique, and it can give us, like, it can reduce the dimensionality. And technically, UMAP is not a clustering technique, but we can use other methods to visualize things as well. So this proposed method is not dependent on any particular clustering algorithm, but any clustering algorithm may be used with Mapper, so that's an important point. We implement this method and present a few sample applications, in which simple descriptions of the data present important information about its structure. So this is sort of the way they approach this problem. They actually say that it's useful to label the nodes of a simple complex by color and size. So you can see that they have this line, and this actually is reminiscent of one of the hyper network models we made for cyanobacteria, where you have this chain, and the chain expands, and along the chain you have these breaks where you get differentiation versus things that just kind of get pushed in either direction. And that might actually be an interesting model system for this as well, where we have very simple morphogenesis that can be modeled with a very simplical set of simplical complexes. So we're labeling the nodes of the complex by color and size. The color of a node indicates the value of the function f, red being high here and blue being low, as a representative point in the corresponding set of cover U bar, or perhaps by a suitable average taken over the set. So we have this, we have the simplical complex. It forms this, line, or I guess, a positive half line and negative half line. But this is something that, we can use in morphogenesis. We can identify simplical complexes associated with certain modes of morphogenesis in different organisms, model organisms, and so forth. So they talk about the implementation of a statistical version of Mapper, in which we developed from point cloud data. So we actually have a lot of point cloud data in DevoWorm. We do, there's cell tracking data, which I'll talk about in a minute. But this is also something that, if we wanted to apply a data set, we're going to have basically point clouds. So if we segment a bunch of cells, the cell centroids are going to be point clouds. We might have features that are point clouds, or maybe the edges of cells are going to be a series of points, and they'll form this point cloud that we have to then find the topological structure. The main idea in passing from the topological version to the statistical version is that clustering should be regarded as the statistical version of the geometric notion of partitioning a space into its connected components. So this is kind of like in network science, where we try to find like the giant component, where we try to find like communities. We try to find the structure within this hairball of connected elements. And so they talk about, we assume that a point cloud contains endpoints that are contained in this set X, and then we have a function from X to R, which maps it to R, whose value is known for n data points. We call this function a filter. Also, we assume that it's possible to compute interpoint distances between the points in the data. So we have this, technique which we kind of use informally for, analyzing point clouds and finding structure in them. But this is actually a more formal method. Now, TDA is not necessarily closely related to network science. So TDA is its own thing. It's based on all sorts of different types of data sets, not necessarily network data. But we can use it in networks very well, and it gives us a lot of interesting information. And so this kind of follows from Himanshu's work last summer, where we have this ring, or we have these structures where we try to decompose the ring and try to find the network in it. So this is where we're sampling from a noisy circle here, and we're setting up our filter, and we end up with this, basically the simplical complex, the six-sided shape. And so you can see that you have actually 8 nodes within that, but it forms the shape of a hexagon. So this just tells us how we do this. At the bottom is a simplical complex, which we recover, whose vertices are colored by the average filter value. So some of these are going to be, have different filter values, blue being low, red being high. So there's going to be different confidence across the structure, but we can at least say that this is a simplex that, has some statistical support. And so although the heuristic has worked well for many data sets that we've tried, it suffers from the following limitations. One, if the cluster has very different densities, it will tend to pick out clusters of high density only. So if you have more data sets or data points in an area, it'll pick from that disproportionately and give you probably what would be the wrong answer. The second case is it is possible to construct examples where the clusters are distributed in such a way such that we recover the incorrect clustering. So, this is where the clusters are, distributed in a way that's going to lead to false positives. And so the procedure needs to be worked out. I'm not really sure the state of the art right now. I'm sure they've been working on these points. They're actually using a sort of a k-means analog here. So increasing k will increase the number of clusters we observe, and decreasing k will reduce it. So this is the heuristic that they were referring to in this part here. But this is, of course, analogous to k-means. So this is something that, again, we're using this in the other techniques. So maybe there's some connection there. We have to find out more. And so finally, we have this high dimensional parameter space that we try to reduce. And so we're doing this data reduction. In general, the mapper construction requires as input a parameter space defined by the functions, and b, a covering of the space. So we need to be able to cover the space, but also define the space. Any covering of the parameter space may be used. So this is a very general technique. It can have a lot of, we can apply a lot of methods to it. And so I think combining the neural developmental programs with the mapper algorithm actually works out pretty well. I know that Himanshu didn't get a lot of chance to work out the details of the mapper algorithm with respect to data, but this is something that, again, we do a certain amount of work in the summer, and then we improve upon it the next year and the next year, and we try to, by the end of the, maybe 4 or 5 year period, we have some really good work. And so finally, I want to reiterate this aspect of the cell tracking challenge data sets. So this is something Himanshu brought up in the conversation, and this is the cell tracking challenge, 10 years of objective benchmarking. So when we're looking for data sets, we can go to the cell tracking challenge, and they have a lot of data sets. This is the paper from Nature Methods from last year where they described the project. They described what was going on in this 10-year period, and they've been generating a lot of training sets for different models. And the idea is to basically do the best job you can in terms of tracking cells in an embryo. So you have these fluorescent markers, and you're tracking those fluorescent markers. You're tracking cell division, you're tracking differentiation, you're tracking migration, you're tracking their 3-dimensional position. And actually, their 4-dimensional position, because those 3-dimensional positions move over time. And so we have all this information that we can extract out of microscopy images. And so speaking of which, we have some microscopy images here. And so these are different data sets where, we have these 3-dimensional images of different systems. And so there's, they usually use some sort of fluorescent protein where they stain different parts of the cell. So in this case, this is GFP labeled actin. These are lung cancer cells embedded in a matrigel matrix. So you can see the outer parts of the cell with the actin in it, and you can see like the structure around the membrane and things like that. We also have lung cancer cells, human breast carcinoma cells, and then of course here is the C. elegans developing embryo. And this is based on data from the Waterston lab, which the Waterston lab has produced really some of the foundational data available for C. elegans embryos. It's very reliable. They've basically, labeled each cell, and they have a time series. So these two data sets, the test and the training data, these are very standard data sets, very well characterized, and they should be of use to like getting the centroid of each cell. And the label is very precise, and it usually has very little noise, although there is noise in every data set. You can see that it's producing, from very, very few cells to a lot of cells, and they're very clear in terms of the background and in terms of the signal. So that's good. But of course, there are other data sets. We have the Drosophila melanogaster embryo, which is the fruit fly. This is a different mode of development than C. elegans. I mean, they have a similar type of development, but it's a very different system. So there are a lot of things going on in this embryo that are different. But this is another data set from the Keller lab where they have worked out some of the details of the data. It's very low noise. They're from Banchalia, so they have like a lot of money to put into this. They have the test and training data sets for this, and this is another data set. There's also a data set for zebrafish from the Keller lab, and I'm not sure if it's on this list, but this is something we've worked with in the past. So yeah, I don't think it's here, but we do have access to other data sets. We have the Systems Biology Institute at Riken, where we also have a number of data sets that we can get access to as well, and that includes that zebrafish data set from Keller. So there are a lot of data sets you can use. Now, development, in the model organisms has been pretty well characterized with these cell tracking data sets. What our challenge is to take those data sets, take the information about spatial position, about the markers and things like that, and use it on some of these techniques, use it for theoretical questions or for interesting analyses. So that's where we're going with this. So that's your update for this week. Next week, we actually have our 10th anniversary of DevoWorm. We've been around for 10 years, and I have a presentation that's going to be on our YouTube channel, hopefully by next week's meeting, but technically the 10th anniversary, I think, is April 11th. And if you watch that presentation, you'll find out why that is. And so look forward to that, and look forward to GSoC, Google Summer Code. We'll be working on it this summer. Again, our chosen students will be revealed at the beginning of May, so stay tuned for that. Hopefully, people who want to work on this project, you don't have to be part of GSoC to work on it. We can develop a project around your own needs. We've done this for a number of postbacks, graduate students, people just interested in biology research and so forth. And we have a lot of people who have really cut their chops on machine learning in our group. So this is something that we also, there are theoretical aspects to this as well. So you don't just have to focus on building software or, being a real expert at machine learning or any associated area. You can do things like work on theoretical questions using some of these tools, but also, even using the data sets. We've done this as well. So if you're interested, let me know. Thank you, and see you next week.